3D proteomics of single cells through molecular pixelation. And the three-letter acronym we have is MPX. So if you hear me talking about MPX, it's just about the technology. My name is Louise, and I'm the field application scientist at Pixelgen. So in short, my role is to make sure that everyone is successful in applying the technology. Great, so 3D proteomics of single cells, what is that? Well, in our technology, we use DNA-encoded barcodes and next-generation sequencing. So there is actually no instrument involved. It's just a reagent kit. You have it here on the counter in the front. And we're using this to assess abundance and spatial organization of single cells on the cell surface for the proteins. So we're assessing the cell surface proteome. What you can see here, this little spinning animation we have, is a Raji B cell. And this is actually an output from the software. And you have CD54 colored in pink. And as you can see, it's interesting because this protein is really clustering to one end of the cell. And this is a cancer cell line, a B cell cancer cell line. And we don't see this with unstabulated PBMCs. So this is something that is specific to just a couple of cancer cell lines. Just a side note, but I think that's really interesting. We'll get more to that. So why is it so important to assess the cell surface proteome, and how can it be so closely linked to cellular function? We have some examples in this slide. The first one being communication and how a T cell is interacting with an antigen-presenting cell. They form something called the immune synapse, which is a very specific protein constellation on the cell surface. And only once it's formed, they can communicate in this way. The second would be an action example and how a CAR T cell is interacting with a cancer cell. It needs to reorganize some of its cell surface proteins in order to perform this perforin granzyme-based cancer cell killing. Third example with attachment and movement or migration. We know that when T cells migrate, they tend to form something called a uropod, which means that some of the proteins on the cell surface relocate to the back part of the cell. You see it here colored in purple, which makes the cell more rigid and more effective in cancer cell killing as the tumor microenvironment is super packed. So it makes it easier for it to migrate. And then the fourth example is an antibody drug mode of action. You have rituximab here colored in pink. What rituximab does is that it binds to CD20 on B cell cancers, which then clusters CD20 to one side of the cell. And only then can NK cells recognize and bind to them to initiate the ADCC pathway. So these are some examples when the spatial aspect is actually crucial. But let's talk a little bit more about the technology. So how does it work? And we'll also go over a short overview of the protocol. In this slide, we have well, we call it a technology overview, but it's also somewhat of a protocol overview. The first step is to fix the cells. And we're working with cells in solution. So we're not working with tissues, but we're working with cells in solution. So we fix using PFA. And the reason for doing that is because we want to immobilize the cell surface proteome from the time of interest so that nothing changes throughout the protocol. The next step is the AOC binding. And AOC stands for antibody oligoconjugates. And it's basically barcoded antibodies. So we have an 80-plex panel in the product, which then binds to the respective targets. And at that point, then we get the abundance information. But then we have two steps of pixelation steps, which are actually where the spatial aspect comes into play. In pixelation A, we generate protein neighborhoods. And in pixelation B, we will spatially connect these protein neighborhoods into one global surface proteome map. I'll talk more about how that works in the next slide. But then we also have more of a standard NGS library prep, such as PCR, beads cleanup, and so on before it's time for the sequencing. And sequencing is typically carried out by Illumina machines. We have NextSeq, NovaSeq, all been working very well. And then comes the data analysis. And for data analysis, we have a software that we've developed called Pixelator to process the data into its correct format. And then we have tutorials handling all of the secondary analysis to really tailor it to your specific scientific question. But we'll get to that as well. So a little bit more details of how this actually works. How do we go from antibodies to spatial protein networks? So if we look at the top left, I think I will actually use my pointer here. Like you saw, we have these barcoded antibodies that we call AOCs that bind to their targets. And at that point, we have surface protein information. So we have the abundance information, but we don't have any spatial information yet. And as you can see, we have this little oligo conjugated to the antibody, which has a unique molecular identifier, as well as a protein barcode. So the protein barcode would then be CD3, for example, for a T cell, but the UMI will always be unique for each antibody. But then we have the first pixelation, Pixelation A. And here we have a reagent that binds to a couple of antibodies that are close to each other and gives them a neighborhood barcode. So you can see that all of these 4 antibodies got the same neighborhood A barcode. These got the same neighborhood A barcode, and these got the same neighborhood A barcode. So you can see how we're really building the amplicon on the oligo of the antibody. And at this step, we get these protein neighborhoods, like I said, because we know they were close because they have the same neighborhood A barcode. But then we repeat that another time with Pixelation B. By statistical chance, it won't bind in the exact same pattern. So then we have, for example, one of the B reagents that have bound to these 3 antibodies because they're still close, just another pattern, and giving them the same neighborhood B barcode. So you can see how we have the UMI, the protein barcode, the neighborhood A barcode, and then adding the neighborhood B barcode. And this will then in silico be able to connect everything into a protein network where you can both see how much you had of the proteins, but also where they were. Right, but of course, this is a new technology. It takes some time getting used to. So if you want to learn more around the details, please check out our paper in Nature Methods that really goes in depth how everything works and all of these barcodes that are being extended. It's also an open access paper. Moving on then to the molecular pixelation data. What are the results that you can get with it, and what does the data type typically look like? As you saw in the former slide, the last step kind of from the lab is the next generation sequencing, typically done with Illumina. And as you know, you're the professionals on that, but you have the FastQ output from that. And the FastQ output is what we import into the software that I mentioned, Pixelator. Pixelator is a free software that we have developed. It's an open source project, so it's available already, but it's aimed for MPX data processing. So it's going to filter the reads, it's going to do some of this analysis, and it's going to put it in the correct format. And the output from Pixelator, there are 2 outputs. The first one is a web QC report. It's going to tell you, was the sequence deep enough? Is this run successful, basically, all of those details. And then the second is the pixel file. And the pixel file is really where the magic happens. That is what you use in the secondary analysis to tailor the analysis to the scientific question you had in mind when you designed the experiment. So to really answer the purpose of why you run it. And then just showing what that can look like. So you have a UMAP. I haven't written all of the different cell types here. We will come to that later. But as you can see here, you have a UMAP here where each dot represents a cell. belonging to a certain cell type. This is a PBMC, unstimulated PBMC is what you see here. And then we can choose one of these cells. So say that we chose this one, and then we can zoom in, it's a memory CD4, and then we can really see how much of the protein and where was it located. And here we're showing CD4 in pink, but it's that is just one of the proteins that you could be looking at. You could be looking at CD3, CD2, or whatever protein that you want to look at. Another cell that we could look at is this one right here. It's a CD8 naive T cell instead. And here you have an example of coloring it with CD8. Talking about the hallmarks of MPX, then we like to divide them into 3 sections. So by now you know that we're looking at the cell surface proteome. And for this, we can really assess abundance. We've already talked about that. But then we have 2 spatial metrics. So it's the polarity and the colocalization score, or the colocalization. And if we start by looking at abundance, then you can ask these more typical questions, maybe, what is the relative abundance of each of the markers in my samples? What markers up or down regulate in the conditions that I have, and to what extent? And that's kind of what you see here in this heat map. You can see how things are changing between different conditions. But you can, of course, also do more UMaps, cell annotation, immunophenotyping, and monitoring if that's more what you're doing. And here you have kind of the same UMap that I showed in the other slide, but here you can also see the annotations. So you can really do more of this deep phenotyping and also subdivide them, choose what cells you want to look at further for the analysis. Maybe you're just interested in a particular cell type. But then I want to show a correlation between MPX and flow cytometry. So what you can see here, you have CD25 here in the bottom, and you have resting T cells and activated T cells. And you can see that CD25 is barely expressed in the resting T cells, whereas in the activated T cells, you can see that it's very much expressed. And you can follow how this works, both with flow cytometry, but also with molecular pixelation. But the first spatial metric then, the polarity. We've developed something called the polarity score, which, if centered around 0, it means that it's randomly dispersed. There's no typical pattern to this protein. It's just randomly dispersed on the cell surface. Whereas if it's positive, it means that this protein is clustering either to multiple poles or clustering to just one end of the cell. And then we can see for each of these markers that we have in the panel, is the spatial distribution of this marker non-random? And if it is, how clustered or polarized is the marker? I'll also do this kind of differential analysis, so seeing what markers increase or decrease in polarity in my conditions, and to what extent. So that was the first spatial metric, and just wanted to show you kind of a proof of principle, what that looks like from the lab. If you remember from the beginning of my talk, I showed you this figure that you have now in the bottom right corner with the example with rituximab, and how rituximab binds to CD20 to cluster CD20, which enables NK cells to bind efficiently. So that's a perfect proof of principle, right? And what we did is we treated cells then with or without rituximab, and we wanted to check in the microscope, of course, to make sure that it actually worked. We could see here that the control samples randomly dispersed all over, whereas the stimulated ones, then it's polarizing to just one end of the cells. And then we ran that through molecular pixelation. And what we can see here is, well, the same thing, but if we just look at these dots here, each dot represents a cell. The blue one is the negative control, so without rituximab, and the red one is CD20 with rituximab. And as you can see, for CD20, it's kind of centered around 0 with a negative control. But once treated with rituximab, you can see how the entire population of cells are actually increasing their expression, increasing their polarity for CD20. And of course, we have heterogeneity when we're working with cells, but you can see how they're all increasing their polarity score for CD20 and clustering to one end, whereas CD45 is still centered around 0. But then the second spatial metric, the colocalization metric. So if we're looking at the bottom here, we've also developed a score for this, of course, in order to assess. And then we have, it's called the colocalization score. And if it's negative, it means that these two markers are actually segregating away from each other. So it's not random. They don't end up next to each other. They're segregating. If the colocalization is centered around 0, it means that it is random. Whereas if it's positive, it means that these two proteins tend to end up closer to each other more than what you expect by random chance. And then the higher the colocalization, the more colocalized they are. So it's also an absolute metric in that way. The same kind of things can be answered with this. Then how colocalized are each marker pair? What marker pairs aggregate or segregate in my conditions, and to what extent? And to show you more of an example from that, we have this little illustration here. So this is then the plasma membrane, and you can see here that this would be a multi-protein domain, so more than just 2 proteins. But you can also have the protein complexes where we have an example of LFA1, which consists of CD11A and CD18. The reason why I'm mentioning these is because we have them in the panel. So here you have this huge heat map of the colocalizations. So you can imagine, this actually isn't all of the markers even, but you can imagine that you have the markers here, and then you have markers here, and then you can check colocalization score for each of the pairs. And what you can see is that we have LFA1 right here, where CD11A is colocalized. So the darker the red, the more colocalized to CD18. But we also have, for MHC class 1, we have HLA ABC in the panel, as well as B2M, where B2M is a subpart of MHC class 1. And you can see how it's same thing, clearly colocalized to B2M and HLA ABC. We also have a third kind of positive control for that with VLA4 consisting of CD29 and CD49D. But what you can see here, so that would be just looking at protein pairs, but we're also looking at protein groups. So you can see that we're able to pick out these kind of multi-protein domains where it's a group of proteins. And sometimes we see these signatures that once treated with something, we could see a migratory pattern, for example. And then we're looking at multiple proteins and not just pairs. So that is something that we think is very important to also look at more of those groups of proteins. I want to end with talking a little bit about applications, just an overview, as well as the product. Here I have some examples of what people have used molecular pixelation for. It's always good to hear a little bit about what people use. So if we look at the first section, which is drug development and pharma, we can see that analysis of mode of action for different small molecules is one of the facts where people have compared different compounds and seeing which one has the most effect. But also the analysis of mode of action for antibody drugs, where we saw the example with rituximab. One very big field for us is the hematology field, where blood cancer research is perfect because we're working with cells in suspension with, for example, ALL and AML, where you can look at treatment response, drug resistance development, and so on. And what we see there is this maybe bigger studies where you know the treatment response. So if you can then take this, if you can use the samples before they were treated, can you predict treatment response? Because that would be the key, right? If you could just take blood from someone and see if they will respond well to treatment. So that is something that a lot of people are investigating. And then it's PBMC or bone marrow samples used for that purpose. Immunotherapy and cell engineering, also very commonly used, doing CAR-T analysis. How can we make them more efficient, CAR-Ts, for example? When do they become exhausted? Why do they become exhausted? Can we find these kind of patterns or signatures of exhaustion, for example? Also treatment response there, looking at checkpoint inhibitors. Instead of animal models, then using organoid models. Also doing more time studies. So we have this T-cell therapy time studies that people have been doing and seeing how things progress over time. So you can really see how it increases over time. And then it's been PBMCs or cells dissociated then from fresh tissue. So not using the tissue itself, but dissociating the cells. And of course, general immunology, looking at more T-cell migration, understanding differentiation, activation, exhaustion, and also B-cell profiling that we will hear more about later. So these are just some examples of what people are doing out there. This is the final slide, and it's just something that I would like to, for you to kind of remember molecular pixelation by. So we know that we're working with cells in suspension. They can be either fresh or cryopreserved. And we're looking at protein abundance and spatial distribution of these 80 cell surface protein markers that we have. And the antibodies are part of the kit, and they have been very extensively validated. So we want to make sure that they work, and we are sure that they work, and they're in the correct concentration and everything, so you just have to pipette them in. The sequencing-based technology, we know that already, and there is no instrumentation, so there's no specialized instrumentation. Sorry if I'm covering here. It's just a reagent kit with a free software. So it's very, very easy to get started with. And you, as I mentioned, you have the kit there on the counter. The protocol is also very simple. It's basically a set of pipetting reactions. So you incubate it at some time for some temperature, then you wash it, you centrifuge, and do a set of repetitions of that. And you can go from live cells to sequence-ready library in 3 days. It's also a very rich data set. You actually get more than 3 million informative data points per sample. And what I mean with informative data points is it's either an abundance, a polarity, or a co-localization metric. So you can really use it for your analysis. And we have up to 1,000 cells as output. And yeah, you know that already, but it's a free software. There is a lot of documentation out there. You have tutorials to help you with the analysis, but also a team of field scientists who are here to support for that. All right, great. With that, I'd like to thank you so much for your attention, and I'm happy to take questions.
Formaldehyde fixation.
Yes.
How much effort does one need to put into optimizing that to get under versus over cross-links and get it right?
Right, great question. So I'm just going to repeat for the ones on Zoom. Talking about PFA fixation, how much optimization is required. We have done that. It's part of the protocol. So it's already a written protocol that you can follow. It's 1% PFA. You incubate it for 15 minutes. But yeah, it's part of the protocol.
Cell type, no optimization.
Yes, that works for all cell types. Yes.
If I can have another one. 1,000 cells, how many reads, sequencing reads would you need for that
Right, yeah. So how many sequencing reads that we need for 1,000 cells? What we have in the user manual is 120,000 reads per cell. So it's quite a lot of sequencing, and it's because we have this rich data output. So it's a deep sequencing with a very rich output. So you get a lot of information from each cell. Yes, please.
You showed us this for single cells. Could it also be used in fixed tissues? Reason I ask is why we are collecting tissue from different autoimmune diseases, from the thyroid, from pancreas, from synovial in rheumatoid arthritis, and it would be nice to see whether there are similar. What are the similarities and differences?
Yeah.
So yes. Can you say something about the tissues? We have formalin fixed tissues, and we have slab frozen.
Right.
Can that be used with this technology?
Great question as well. The question is regarding if we can use fixed tissue for molecular pixelation. And we cannot do that at this stage. We're working with cells in suspension only. So if using tissue, it needs to be dissociated once the tissue is fresh. And the reason why we're, of course, looking at ways to do this, but the reason why we cannot do it right now is because that fixation is so harsh. So it's not really working well with the antibodies and getting these protein neighborhoods and all of the spatial aspect. So not at this moment, but we would love to get there, of course, as well to do that. Yes. Oh, we have the microphone now. Perfect.
Thank you. Thank you for the presentation. So I have a couple. So the first question is, you have only surface proteins, so there are no intracellular proteins, and there are no plans on intracellular proteins?
Yeah, no, right. What you're saying, we only have them on the cell surface right now. And we don't have any intracellular markers currently, but the same thing as tissue, we would of course like to go there someday. It's just not been a priority at this moment, but.
Yeah, I see. And the annotation part, so just like cell type annotation, how is it done? Is it just like automatically done, or one can just like influence it? Because, you know, we would like to study some rare populations. I mean, that not very predefined, and generally immunology probably moves towards that we skip these just like the names, generic names for the cell types, and rather call them just like by expression markers. So how the annotation is done?
Right, yeah, so we are doing the annotation manually because we have found that that is the most accurate way of doing it. We have looked into automated ways to do that, but it's so different. Some people run PBMCs, some people run selected cells or cell lines. So we rely typically on the experts doing the experiments themselves for the annotation.
Okay, so I basically get my data and annotate the cell types by myself manually. Okay.
Yeah,
okay,
that
was
is
my question.
we have found to be
Yep.
the most
Okay.
efficient.
Yep. Thank you.
Yeah, thanks.
So we have one question online. I will read it to you.
Yeah, thank you.
So is it possible to trace a molecule's journey in a live cell, not fixed cells?
Right. The problem with that, if we were to use live cells, is because throughout the protocol, there is so much time, there is time, and there are different temperatures, there are different buffers, and this is going to affect the spatial aspect of the proteins. Even abundance can be affected by that. So that is why the first thing is to fix them. But I think the question is more, okay, so the thing is that you do your experiment, you can do your stimulations or thaw from patient samples, and then once you have them at your time of interest, you fix them. Because I don't see why you would like to know how it's affected by the protocol, but because that would then skew the data. It wouldn't be the right data from the time of interest. I hope that answers. Feel free to ask follow-up questions otherwise.
So I guess if you're interested in a time lapse, how a protein is changing, you could also fix it at different time points.
Yes.
So have a simulation that goes on at different time points.
Right, so then you take cells out at different time points, you fix them, and then you freeze them. So you can freeze the fixed cells so that you can thaw everything at the same time and run everything at the same time. Yeah, that's probably what they meant.
Great. Do we have any other questions from the audience? No? Then I think we thank Louise for that introduction to the technology.
Yeah, thank you so much.